BioCyc reaction pages provide detailed information about one biochemical transformation. One way to get to a reaction page is by clicking on a reaction arrow within a pathway page. In this case, the E. coli homoserine biosynthesis pathway. Moving down the page, we have the title for the reaction, which will be its EC number if one is assigned. Then the superclasses area shows one or more ontology classes to which the reaction is assigned. Next, we see that 3 isozymes catalyze this reaction in E. coli, and that experimental evidence exists for the function of each enzyme based on these flask icons. And we see the genes that encode each enzyme. Further, this reaction is present in 2 different metabolic pathways. Next, we have the reaction diagram. Notice that conserved moieties in each compound are colored using atom mapping information. And by the way, note that this and related features are available only in some web browsers. So it's the green conserved moiety that's visible or not. We can turn the coloring on and off. And we can also add a number to each atom if desired. The numbers are the same on both of a given atom are the same on both reaction sides. Also, mousing over an atom shows its corresponding atom on the opposite side of the reaction. Now, by default, reaction direction is that assigned by the enzyme commission, which may differ from the physiological direction of the reaction. Note also that enzyme-specific information about the reaction, such as kinetic data, are available on protein pages. The gene reaction schematic is a diagram that depicts the many-to-many relationships that can exist among genes, their protein products, and the reactions that they catalyze to make these sometimes complicated relationships more apparent. We can mouse over these entities to see which biological object they represent. The shaded box represents the page we are currently viewing, this reaction page. The 3 circles represent the protein complexes that catalyze this reaction, and a second reaction shown here. 2 of the complexes are homodimers. The third is a homotetramer, based on these numbers. The 3 purple boxes represent the genes that code for each of those proteins. A number of operations are available for reactions in this operations menu on the right sidebar, such as downloading structured data associated with the reaction, such as atom mapping data, and a list of genes for the enzymes that catalyze this reaction. Note there is a search reactions command that allows you to search for reactions by a number of criteria or to generate a list of all the reactions in this PGDB. For example, we can search for all reactions that have L-aspartate and ATP as a reactant or a product. Let's submit that query, and there's our resulting list of reactions. The next webinar in the series will introduce you to BioCyc metabolite pages.